In this lesson, we're going to look at the history now of how cells were discovered, and we're going to look at types of cells in this lesson as well. So to begin with, remember I told you this first statement right here is a lie. Leeuwenhoek did not invent the first light microscope. What he did was re-engineered light microscopes. This guy, Robert Hooke, actually came first. He, in 1665, actually discovered cells. He discovered and named them. So he was looking at slices of cork, and remember, cork comes from the inside of the bark of a tree. So these are plant cells. And he noticed that basically the cells were little empty boxes, and where he noticed that the cork was made of empty boxes, and he called them cells because they reminded him of the small empty rooms that monks in a monastery live in. And we call small empty rooms cells. That's what you call them in a prison. That's what you call them in a monastery. So they reminded him of those small empty rooms, so he called them cells. Leeuwenhoek actually took the design of the microscope that Hooke was using, and by the way, nobody knows for sure who actually invented the first light microscope. But what we know was the one that Hooke was using wasn't a great microscope. So Leeuwenhoek actually took that design and turned it into what looks more like a regular microscope today, what most of us would recognize as a microscope today. Now, within short order, for science terms, in less than about 200 years, two German scientists, Matthias Schleiden and Thomas Schwann, were able to conclude that all plants and animals were made of cells. So using Leeuwenhoek's re-engineered, redesigned, new and improved microscope, they were able to build upon the work of these scientists and conclude that with confidence that all plants and animals were in fact made up of cells. Now, the work of all of these scientists and all the scientists since then are summarized in the cell theory, which we're going to get to in just a minute. Now, here's the thing. Remember that term cell, which means empty room, right? Until about 70 years ago, remember, a lecture on cells in a biology class would sound something like this. Okay, class, here is a plant cell right here. It's got a cell wall. It's got a nucleus with some DNA in it. There's a membrane inside. I'll draw the membrane. There it is. Here's an animal cell, right? You've got a membrane, no cell wall, a nucleus, and DNA inside. Boom, done. And the reason why that's like that is because we were only using light microscopes until about 75 years ago. Okay, now let's look at how microscopes have changed first and get you an idea of why the light microscope is a problem. So the light microscope that Leeuwenhoek redesigned was called a simple light microscope, and it was simple because you only were looking through one lens when you looked through it. But it is a light microscope because light is passing through a tube that's passing through the lens, and so you're seeing it magnified and lit up, illuminated. The problem is Leeuwenhoek was grinding his lenses by hand. You know, we're talking about a long time ago. There weren't computers, there weren't lasers, there weren't the kinds of technology available that make our lens grinding nowadays really, really accurate. So Leeuwenhoek was grinding his lenses by hand, and so the magnification was really low. Since Leeuwenhoek, we've actually improved upon the microscope design even further by creating what we call a compound light microscope. And it's compound because anytime you're looking at something under a compound light microscope, you're looking through two lenses at the same time. So not one lens, but two. The reason why that's significant is when you look through two lenses at the same time, you actually, under the laws of physics, multiply the magnification of both of those lenses, the eyepiece and the objective lens, to get the total magnification. So you're looking through that little eyepiece, that's got a lens there, and then down at the bottom there's another lens, that's called the objective lens, and you multiply those magnifications. So you get a lot more magnification just by adding a second lens. So we get a lot better magnification. Obviously, our technology is much better. So we've got high quality lens grinding, so we can make our magnification much better. But light is still passing through a tube. Okay, so while we can get up to about 1,000 times magnification, we can't get much further than that. And the reason is that when light passes through a medium like air, it bends, right? So, and you've seen this when you've stuck a straw in a glass. You can see the light bends differently in the water versus in the air, and so it looks like the straw is kind of broken, right? So that's because of this light refraction or this light bending, okay? What that means to us is it provides distortion in the image. So our image is not sharp, it's not clear. So with a light microscope, when you look at cells, you see little more than what we were just talking about here at the bottom, okay? So the cell used to be referred to as the black box, and the reason why it was called the black box was because we knew there was a lot going on there, but we really couldn't see it. Even though this was all clear, and you could see through it, you couldn't see the individual organelles that I know, you know are there, right? So it took the invention of the electron microscope for us to be able to see all the itty-bitty bits and pieces inside of cells, all those little tiny ribosomes, which are the smallest organelles, all the endoplasmic reticulum and Golgi and everything else that you're used to hearing about. The reason why electron microscopes work so well is instead of light, they use a beam of electrons. So no light involved, which means no distortion. And those beams of electrons are shot through a vacuum tube, so there's no medium through which it's passing to bend the beam of electrons in any one way or another. These these electron microscopes are so good. Some of them can even see the electron cloud of individual atoms. So you're talking about big time magnification, which totally kind of made the whole world of cells explode for scientists within the last 75 years or so. All right, I promised we'd get back to the cell theory, and here it is. Now, to you, this seems like one big, huge duh, right? Duh. But it was really, really, really revolutionary. Okay, so the work of the scientists, you know, from Robert Hooke onward, is summarized in three very important statements. The first statement is that all organisms are composed of cells, and you know this from Unit 1, because remember, that's one of those characteristics of living things. Second thing is that the cell is the basic unit of organization of an organism. In other words, the smallest living component of an organism is its cell. I could take one cell from your body, stick it in a lab, give it food and water, and it would live on its own. Okay, you can't pull your mitochondria out and expect them to live, right? But you can pull an entire cell out and it can live on its own. Finally, all cells come from pre-existing cells. So cells don't poof into existence spontaneously. They have to come from cellular reproduction. And those are the three statements. Yes, you have to memorize them, but they're not that difficult to memorize once you get what's going on there. All right, so in terms of cells and their types, there are two main types of cells, and this we talked about again in Unit 1, but now we're going to expand on it a little bit. So the prokaryotes, pro means before, karyo refers to the nucleus. In other words, cells that existed before the nucleus existed. They have no nucleus and no membrane-bound organelles. And a membrane-bound organelle would be like your chloroplast, your mitochondria, your ER, your Golgi, all those organelles that are surrounded by a membrane. The eukaryotes have all of these things. Now, these include, oh, and by the way, the prokaryotes, we called those the simple cells, but those are the bacteria. The eukaryotes are larger cells, and they have all that stuff inside of them. So why are they larger? They're larger because they've got all these organelles, okay? And remember, plants and animals are part of this, and so are protists and fungus. So just to give you a really quick overview of cells in general, just a visual of what we were just talking about, okay? Here are our two categories of cells right here. You've got the prokaryotes and the eukaryotes. So all cell types are either prokaryotic or eukaryotic, and the prokaryotes are divided into the archaebacteria and the eubacteria. Bacteria, that's the key there, right? Okay, so these guys are all bacteria. The eukaryotes are traditionally divided into the plants and the animals. However, don't forget that protists and fungus, these guys down here, are also eukaryotic cells. We're going to talk about plant and animal cells in a minute. Those are exemplar cells, okay? But fungus and protists are also eukaryotic cells. And in the next unit, we're going to talk about how they're modified a little bit differently. All right, finally, the last thing I want to do is just touch on what the difference is between a plant and an animal cell. Okay, the first thing is shape, right? Animal cells are round and irregular. Right? So the plant cells tend to be boxy or square, and they have these regular shapes. And the reason for that is the cell wall, which only plant cells have, right? Okay, animal cells lack a cell wall. There's nothing there, no cell wall. Now be careful, both have a plasma membrane. So let me draw a little column here. Both of them are going to have the plasma membrane or the cell membrane, okay? It's just that plants have the cell wall and the cell membrane. Animal cells have just the plasma membrane. Also, chloroplasts are present in plants, right? And that's because of photosynthesis. The chloroplast is the organelle that's going to do photosynthesis, and plants need that. And I apologize, I'm doing this with my finger because I forgot my stylus today, so I apologize for the handwriting. All right, again, animal cells, no chloroplast. Okay, so those are the main differences. The last thing is when we get to cell division, this is going to be important. Animal cells have what we call a centriole. Okay, plant cells lack the centriole. There is no centriole. And again, right now we're not going to worry so much about that particular organelle, but in the next unit we are going to look at that organelle. The centriole is the only organelle that exists in animal cells and does not exist in plant cells. Otherwise, all cells have all the other eukaryotic organelles. Okay, so otherwise you're going to see ribosomes, you're going to see ER, you're going to see Golgi, everything else is present in both plant and animal. Last thing that is different is that plant cells have a large vacuole. And they have this large central vacuole, right, for storing water and enzymes. Animal cells have a vacuole, it's just that theirs is small. And I'm going to just put small there, understand that small vacuole, but my face is kind of covering where I would write that up. But anyway, you get the idea. Small vacuole in animal cells, large vacuole in plant cells. So that just kind of summarizes our understanding of cells. Don't forget to study those organelles, because you need them, right? And that's kind of we're now ready to go on to the plasma membrane.